Okay, so this is an overview of the vocabulary in Chapter 11, Mendel and the Gene Idea. So our first word, you can go ahead and feel free to take your own notes as you watch this. So it's a test cross. So let's say in dogs, having one spot is the dominant trait to no spots. So a dog who has one spot would be homozygous dominant or heterozygous, and a dog with no spots would be homozygous recessive. So let's pretend that I find a dog on the street and I'm like, oh, cool, this is an expensive dog. I want to breed it. Well, I can see that it has one spot, but I'm curious. Is it homozygous dominant or heterozygous? Because I know it's phenotype, so I know it has at least one dominant allele, but I don't know its genotype. So what we can do is called a test cross, and that's where we cross the individual with a homozygous recessive. And with that, we can look at the offspring. So over here, you can see the parental generation. So here I have like the P stands for the parental generation. Right now, this dog is, I know it has one dominant allele, but I don't know what the second allele is. So I test cross it with a recessive individual. And so I know that this dog I know this dog is recessive or homozygous recessive because it does not have any spots, and that's the only way it can have this phenotype. So here, if I check out all their puppies, and oh my gosh, every single puppy or offspring has spots, then most likely that parental dog I found is homozygous dominant, and therefore all of the F1 are heterozygous. Now, on the opposite, if they have some offspring that are some with spots and some without spots, well, then I know that that parental dog I found right here, this one, would have to be heterozygous because it has to give one of the recessive alleles to this puppy in order for it to be homozygous recessive. So using a test cross, you can tell it's basically used to figure out an unknown genotype. So anytime you're reading a word problem and it says test crossed, it means it's crossing it with something that is homozygous recessive. Now, you also see words monohybrid crosses and dihybrid crosses. So Mendel did experiments crossing a single character or trait. So when he used his like purple and white flowers, he worked with true breeding purple flowers that were homozygous dominant, and he crossed them with true breeding white flowers that were homozygous recessive. And all of the progeny means offspring, were hybrids for one trait, or monohybrid. So here he had pure breeding purple flowers, pure breeding white flowers. All of the F1 were heterozygotes or hybrids. So when you cross two F1s, these F1s are hybrids for one trait or monohybrid. So crosses with two heterozygotes like this are called monohybrid crosses. Okay, and then you have dihybrid crosses, and with a dihybrid cross, Mendel figured out the law of independent assortment by working with two traits. So if he worked with seed color and seed shape, he found that when he crossed them, this showed up a little late there. He found that when he crossed these like two traits, so you can see here how seed color and seed shaped, green or yellow, round or wrinkled, are two totally different traits. You get here, that would be a hybrid for two traits or a dihybrid. And when you cross a dihybrid with a dihybrid, you get a phenotypic ratio of 9 to 3 to 3 to 1. And so using a dihybrid cross, you can see if the traits stay together as like a package deal or if they inherit independently of each other. Now we have what's called some exceptions to the rule of complete dominance. So when we talk about complete dominance, we're talking about brown eyes is dominant over blue eyes. Purple is dominant over white. But there's some exceptions. So here with incomplete dominance, neither allele is completely dominant over the other. So the F1 hybrids have a phenotype somewhere in between the two parents. So if we take snapdragons, for example, you could cross red and white snapdragons, and you can see how all of the heterozygous are pink. And when you cross, you do a monohybrid cross, and you cross two pink snapdragons, you can see down here how you would have a 1 to 2 to 1 phenotypic and genotypic ratio. Now, if you you do a test cross, you take an F1 monohybrid and you cross it with a white homozygous recessive, you can see you get a 1 to 1 phenotypic and genotypic ratio. And then there's something called codominance. And this is when the two alleles affect the phenotype in separate, distinguishable ways, meaning there's not a blending of traits. So in codominant, this is made up here, but it's like if you had a red fish and a blue fish. In the offspring, you could see both red and blue. If this was incomplete dominance, the F1 progeny would be purple. And they're not purple, they're red and blue. Together, they're both showing up in the phenotype. Now, when we look at our next thing, we're going to look at multiple alleles. So Mendel got really lucky in studying the plants. For each of his traits he studied, they only had 2 alleles. For the flowers, it was purple or white. For the height, it was either a tall plant or a short plant. It was yellow or green, round or wrinkled. And so for his traits, there wasn't any other options. And they all followed like complete dominance. Mendelian genetics. There was no incomplete dominance or codominance. However, there's some characters or some traits where there's more than 2 alleles possible. And so for that, we're going to use some cute little bunnies as our example. So here you can see in rabbits, you have the dominant allele, which codes for the wild type. Wild type is generally what you're going to find maybe most likely in nature. It's the generally dominant alleles. So here you have the wild type, brown fur. Then you have chinchilla allele. You have a Himalayan allele. And you have the recessive allele. So even though you can see here there's 4 different alleles, hence the name multiple alleles possible, each rabbit only inherits 2. So multiple alleles means there's multiple options in that population. But like this rabbit here, if it inherited 1 from the mom, 1 from the dad, this rabbit would be heterozygous and have the wild type phenotype. If it inherited this, again, wild type phenotype. If it inherits this combination, chinchilla is dominant over Himalayan, so it would have the chinchilla phenotype. In this combination, Himalayan is dominant over albino, so it would have the Himalayan phenotype. With 2 copies of the recessive allele, this rabbit would be albino. So while there are multiple alleles, each individual only inherits 2 copies because 1 from the mom, 1 from the dad. Now, I do find that students have a trouble with differentiating between multiple alleles and polygenic. So with this, polygenic, let's break the word up. Polygenic means many, and genic is referring to genes. So this is where you have many genes are influencing 1 phenotype. So it's the additive effect of 2 or more genes on a single phenotypic character. So skin color is a perfect example. There's multiple genes that you inherit that influence your phenotype. So if I look at this pretty large Punnett square here are, I don't know if you'd call these tri-hybrids. Not sure. Never seen that written. But here you can see these 2 parents are hybrids for all 3 genes influencing skin color. And so if the individuals inherit most of the recessive alleles, possibly, you can see they're going to have a lighter phenotype versus if they inherit more of the I don't know if it's dominant or not alleles, they're going to have a darker phenotype. So these two parents can have any combination of offspring as far as their phenotypes are concerned. So it's multiple genes, depending on which ones you inherit, influencing the phenotype. And then with eye color is also an example of polygenic traits. I know we teach brown eyes as dominant over blue eyes, but there's different shades of blue eyes, and there's different shades of brown, There's different shades of brown eyes. And so while, yes, the amount of pigment you make influences brown or blue, there's other genes involved with the intensity and how brown or how blue, or are they green or are they hazel, etc. And then our last thing is pedigrees. Now, pedigrees are tree diagrams that are going to describe traits of parents and children across generations. So, for example, this could be like my grandma, for example. My grandma and grandpa, this would be where my mom falls into play, my mom and dad. This would be me and my husband, and then our children. So when we look at pedigrees, females are circles and males are square. When you have a line between them, that means they're married. And then coming down, this represents their children. So in generation 2, female number 1 and male number 2 had 4 kids. They had 2 boys and 2 girls. And when we see these things colored in, what we can see is if it's colored in like this, that means it's expressing whatever trait we're studying. So if I was curious about blue eyes, for example, I could see here that this father was homozygous recessive. And I know that the parents do not have that trait, so they would have to have a dominant allele. But in order to have a homozygous recessive, they would be like this. So I can tell the grandparents' genotypes. And then I know that because they have 4 kids and 3 of them have the blue eye phenotype, that means that this mother would have to be heterozygous. And so she gave the light blue allele to 3 of her kids, but the dominant allele to son number 2 right there. So anyway, in pedigrees, we can follow the inheritance of traits. You could see out of these 4 children, only 1 has blue eyes. So that means that this parent would have to be, these parents would be like this. Oh, look at that 3-to-1 ratio. Okay. All right. So here's another pedigree. Now, when we look at pedigrees, though, we usually talk about the mode of inheritance. Is it being inherited dominant or recessive? Is it sex-linked or autosomal? And so those are questions that will usually be asked when looking at pedigrees. Like, what is the mode of inheritance? In this one, I know that it's a recessive trait because right here, neither parent has it, yet they have a kid with it. That means that most likely they're just carriers and they're carrying that allele with them. And so, yeah, you want to look for clues, but we'll practice that in class more. All right, good job.